So now we go to the third talk of the session, and this is by Lorenz Keil, and he's a student in the group of Dieter Braun in the physics department at the LMU in Munich, and he will be talking about playing evolutionary games, and the title of his talk is Length Selection and Replication in a Thermal Flow Chamber. So the floor is yours.
Thank you very much for the kind introduction as well as the possibility to speak here. So, today I would like to speak about a theoretical and experimental study we carried out with the group of Dieter Braun at the LMU in Munich, which is about the origin of life. So what is kind of understood is how we get from a single cell indicated here to the large variety of species we have today. And the key mechanism is Darwinian evolution, which is the process of selecting the most successful phenotype. And this is done by selection, replication, and mutation. The question, however, remains how something as complex as a single cell has spontaneously self-assessed, especially in a highly diluted prebiotic ocean. And now we shift the question from how to where, so which location enables a continuous Darwinian evolution. In the late 1960s, Spiegelman encountered a problem that's called tyranny of the shortest, which states that the genetic information is quickly lost. And this is because short nucleic acids are replicated with faster kinetics than longer sequences. So during the replication, the mutation can change the length of the sequence. The result is an evolutionary race towards ever shorter sequences. So we not only need an environment that enables continuous Darwinian evolution, but also maintains the molecular complexity here. So when we focus on a non-equilibrium setting here, which is driven by a thermal gradient. So all our experiments here are conducted in water, and we use DNA instead of air, simply because it's much easier to handle, and second, we can use the polymerase chain reaction, the PCR, as a placeholder for a prebiotic plausible replication mechanism. And this temperature gradient here helps us in the case of replication. So at high temperatures, we can denature the DNA, so into single-stranded strands, and at low temperatures, we enable an elongation. And the natural system for such temperature gradients can be found in hydrothermal vents, so in the white smokers and black smokers. So hydrothermal vents emit hot water in the region of about 400 degrees into the cold ocean sea, and so they form a natural temperature gradient here. We now want to mimic such a system, and we call it a thermogravitational core. And this thermogravitational core is just a chamber with a width of about 100 micrometers here, but we can also choose other width here. And so it's a chamber filled with water. In our case, since we work with DNA, it's DNA diluted in water, and now we apply a temperature gradient. So one side we heat, and the other side we actively cool. And this induces two effects. So first we have convection. So based on the thermal expansion of water, the water rises on the hot side, shown here with the white arrows, and moves down on the cold side. And second, we have thermophoresis, so the movement of molecules along a temperature gradient. So for DNA moves from the warm side to the cold side. And the superposition of both effects now lead to an accumulation of molecules. So for example, if we insert a 36-mer into the system, the DNA accumulates here at the bottom of the pore. And if you look at the trajectory, a stochastic trajectory here, it shuffles in the convective flow from the hot to the cold side, but slowly moves to the bottom here. And still at the bottom, we have cyclic temperature change. That's an experiment here. So this is our chamber, which is filled with the DNA, and the DNA is labeled for fluorescent label. And now we apply a temperature gradient from the back to the front. As you can see, within minutes, all the molecules, the DNA, moves to the bottom here and accumulate at the bottom of the pore. and that's against entropic cost. And the accumulation depends on the Curie coefficient. So it depends exponentially on the Curie coefficient, and the Curie coefficient increases with the length of the DNA. So for example, if we change the 36 mer and go to a 75 mer, we almost double the Curie coefficient, and therefore we almost quadruple the concentration here at the bottom of the pore. But that not only works for DNA or RNA, but for a lot of other molecules. So here we have a simulation where we simulate different diffusion coefficients and different Curie coefficients. And the white line here indicates DNA. So here we have about 10 bases, and here we have about 250 bases. And that's the concentration we can achieve in a pore with a temperature gradient of 500 Kelvin per meter, which is prebiotic plausible. And we can accumulate it up to the power of 10 to the power of 60 for molecules with the high Curie coefficient. Now if we now open the pore at the bottom and at the top, we can now add an influx. So in such high thermal vents we have large convective flows. Now with this influx we add two important features to the system. So first we have a continuous influx of molecules, so we can nurture the system here, and this influx itself acts as a selective pressure on trapped molecules. And this is shown here. So we have flow profile, so we have a parabolic influx, and we have a convection flow. And the superposition of both flows lead to an asymmetric flow. And second, here's the concentration profile. So since the longer molecules accumulate better in the pore, they are stronger pushed to the coldest side. So their location is more likely more based on the cold side here of the pore. And if you compare the flow profile with the concentration profile, you can see that the long molecules are now affected by the downward stream. So they are transported into the trapping region, while short molecules will be flushed out of the system. So in the experiment, it looks like that. So in this case, we use two different DNA strands, a 25 mer and a 75 mer, both labeled with a different fluorophore. So on the left side, we have the 75 mer, and on the right side, the 25 mer. The actual trapping region, we cannot image it, but the front and the back of it. And we now apply an influx. And the influx we steadily increase. And as you can see, at low velocities, both strands accumulate, but at a certain point, the short molecules will be flushed out, while the long ones still accumulate. So we have a depletion of the short strands and still an accumulation of the long. And so we have a clear cutoff here. And this cutoff, as can also be seen here, so in this case, we injected a 200 base per ladder, 20 base per fragment, so here 200 bases, and here's 20, 40, 60. We injected into the system, applied an influx of 6 micrometer per second, and we extracted during the experiment the outflow DNA, and at the end of the experiment, the trapped DNA. As you can see, only short molecules, so 80 bases and below, will be flushed out of the system, and even after 6 hours, all longer molecules remain within the trapping region. And this experiment can also be further extended by slowly increasing the influx velocity. So starting with 5 micrometer per second, we can see a depletion of the 20 and 40 bases, and for a velocity of 50 micrometer per second, we can even flush out the 200 bases. And this velocity-dependent trap fracture can also be modulated by a fluid dynamic model, which is shown here. Now in the next case, we add a replication mechanism to the system. So as a reminder, on the left side, we can see the trajectory of a 36 mer, but this time inside an open pore. And as you can see, despite the fact that it accumulates, so it's trapped, it still shuffles between the hot and the cold side. So we have cyclic temperature changes here. And these temperature oscillations are plotted here, so once for a 36 mil and here for 75 mil over the time. And as you can see, based on the diffusion coefficient, the short molecules move more often from the cold side into the hot region than the long strands. And a temperature cycle is completed when the molecule first moves into a high temperature region where it denatures, and afterwards into a low temperature region for the elongation step. And this is marked with the black circles here. So for example, the 75 mil completes a temperature cycle. If it first moves into the hot temperature region, afterwards into a low temperature region, and again into a high temperature region. And since the melting temperature of the 75 mil is much higher than for the 36 mil, it's even more unlikely for the 75 mil to complete a temperature cycle. So in this case, we have about 2 to 3 times faster temperature cycle for the 36 mil. So in this case, it's about 17 seconds for the 36 mil and about 40 seconds for the 75 mil. However, just by conducting a standard PCR inside the chamber, it works pretty fine for all lengths, and it works even faster than in the cycler. So we have faster temperature range inside this pores. Now to sum it up, so in this thermogravitational pores we have an accumulation, so we concentrate molecules, and more specifically DNA, and we have an influx which nurtures the system and also selects for length, and also the temperature gradient enables a replication mechanism. And if you combine all these 4 effects, you can now modulate the outcome of a PCR reaction. And the most interesting regime is shown here. So here's the frequency plotted against the number of cycles. So at a certain velocity, which is in this case 4.5 micrometer per second, the short strand will be flushed out of the system while the long strand still remains within the system. But here we have now a inversion of the Spiegelman dilemma, because now the long strands now undergo more temperature oscillations than the short ones. So in this case, the long molecules will outcompete the shorter ones. If we now, however, increase the velocity to higher velocities, for example 5.5 or 7 micrometer per second, you can see that in the case of 5.5 micrometer per second, both replicate similar often, and at higher velocities, even the shorter strand will outcompete the long strand. And that's just based on the diffusion coefficient, since both are flushed through the system, and the short molecule based on the diffusion completes more temperature cycles while being flushed out. And that's also the case for a low velocity. So the most interesting regime is at a certain velocity, in this case 4.5 micrometer per second, and now we conducted the experiment exactly with these parameters in the thermal pore. And as you can see, if we start with the binary solution, so 50% 36 mil and 50% 75 mil, the 75 mil outgrows the 36 mil. And so at the end, so after 6 hours, only 75 mil DNA is left within the pore. While if we are, for example, conducting a bulk experiment in the thermocycler and starting with 90% 75 mil and 10% 36 mil, the 36 mil outcompetes 75 mil, like Spiegelman demonstrated. So in this case, we have shown that there's in the thermogravitational pore, there's a regime where we can invert the dilemma of Spiegelman. Now, to finally sum it up, so what we've shown so far is that the thermogravitational pore is able to select, in this case, for a specific length of the DNA, and it's also able to replicate DNA. In order to complete the Winium process, we now need to add a mutation. So the next step is to make the replication a mutagenic replication. And I'm now at the end of my talk, so I thank you for listening, and I also thank my supervisor, Dieter Braun, and the whole lab, including Moritz Keil and Simon Lansky, who performed the work. Thank you very much.
Great, so thanks for the talk. The talk is open for questions. Questions?
Hi, could you talk a little bit about the sequences that you used for this?
Specifically, or?
Yeah,
So-
because I remember Dieter did some very nice work on RNA at one time, and when you get to that number 75, it gets to be interesting if you also start seeing the formation of the tRNA.
Okay, so in this case, we chose two lengths which are in the serine coefficient very different to make it more easier for the selection process.
But you didn't care what the exact sequence of
So
the DNA, the nucleotides were.
We just designed them that they do not interact with each other, so no secondary structures. And since the temperatures here are very low, so we have primers of 40 mer primers, the design was very difficult to achieve. That there are no secondary structures, primer arm, or something like that.
Okay, thank you.
So let me continue a little bit on what Zen was raising. Basically, you say you can select for lens, but lens is sort of whatever lens means. Basically, you have two species, one short, one's long.
Yeah.
How about if you want to select for two things that have the same lens? So basically, that's going to be just geometrical, or there's any other thing you can do. So
we're just working on that problem, but it's very difficult to achieve because the rates are.
But if we're going to do mutation, you need something like that, right? Because otherwise, how are you going to differentiate?
Yeah, sure. So that's exactly the problem we are still working on. It's not that.
Thank you for the talk. So the GC bounds are stronger than the TA bounds. So it would be interesting to know the composition of the nucleobases in the end to see how that compares to the short one.
So we
Do you have any idea?
we designed both of them that they have about 60% GC content.
Right, so that's all very interesting. So you can also separate uranium-235 and 238 using the same methods, which is molecular weight dependent, so which goes to diffusion constant. So if you had the same sequence, then it would have to have a different conformation than to give a different diffusion coefficient, and then you could conceivably separate the two.
So in this case, if you want a replication as well, you need to have high temperature regimes, and I guess the secondary structure will then not exist any longer.
Yeah, okay, right. Well, if you're saying, then another question, if you're saying this was related to the origin of life, then you need this defined geometry, to make this work. Are you saying that? And if so, where would that geometry exist in the primordial world?
So the cracks are basically spread in the hundreds of micrometer regimes.
Okay.
And the one we used is just the maximum because we don't want to waste so much time and wait until it accumulates. And such an experiment takes about 6 hours. But if you're in another regime, it takes months or years.
Okay, right, gotcha. So it's a very, very fine structured, long, primordial spaces. It would take, months, years, longer. Thanks.
So in the region of months.
So you compared it to the thermal vents. Have you guys tried it where you're heating from the bottom and you have cold on the top rather than it going horizontally?
If you're heating it from the bottom, then you have a chaos regime. So you want to have an exact starting point.
Thank you.
But you can tilt the whole chamber.
Okay, further questions? Okay, if not, let's thank Lawrence again.